so today we're going to learn how to identify an unknown bacteria, identify what species it is. Okay, so you're going to be using a range of tests to identify a bacteria. These bacteria unknowns have either been cultured from a skin wound or a fecal culture. Okay, so we're kind of doing a make-believe thing. You are going to be getting the results of 3 different unknown bacteria that are either from the skin wound or from a fecal culture, okay, hypothetically. Right, so once you get your 3 tubes of unknown, which you would if you were in lab, you'll have 3 different broth cultures, okay, unknown 1, 2, and 3. They were either taken from a culture from a skin wound or a fecal culture. Okay, so how did we come by having these isolated cultures from these, the wound or the fecal sample? Well, it was taken, let's say hypothetically, by a doctor. The culture was either taken by a swab of the cut, a skin lesion, or something that's been infected. Okay, so it's going to have many different types of bacteria in it. Some that belong there and some that don't. Okay, or a fecal sample that was collected and it was sent off to a lab. Okay, once it got to a lab, they isolated the different species of bacteria that were present in either the skin culture or the fecal culture by using streak plate technique. Okay, you remember a couple weeks ago we learned that using streak plate technique you can take a mixed culture such as something that is taken from a wound, okay, or a fecal sample. You can take that mixture of bacteria and isolate, by using streak plate technique, different colonies of isolated colonies of each of those individual species of bacteria. And then from that colony, you can go on to use it, because it's a pure culture, that isolated colony is a pure culture of that bacteria. You can use that to identify what kind of bacteria it is, since it's a pure culture. Okay, so the hypothetical results you are getting have been taken from either a skin culture or a fecal culture. It is 3 of the unknown bacteria in one of those cultures. Okay? Now, everybody doesn't have the same 3, even if you have a skin culture, okay, or a fecal culture, because I have given each of you 3 different ones. Okay, so make sure you're doing your own work. All right, so just knowing that if it's skin or fecal is going to help you. It is identified in your results, whether you have a skin culture or a fecal culture. Those are important things to know when you answer questions associated with these identifications once you're done. Okay, so how are we going to identify the unknown bacteria? Okay, so we're going to go through how you inoculate all these different media with your unknown number 1, and then you would do the same thing for your unknown number 2 and your unknown number 3. Okay, so you have your culture, broth culture of unknown number 1. Okay, remember it was provided to you by a technician hypothetically, and they obtained it from streak plate technique. So they said, okay, this is an isolated bacteria we got from the skin wound or from the fecal culture, and we need you to identify it. Okay? So if you look at your handout, the first thing you would do is a gram stain. Okay, very important. This is always the first step, because this is concrete for one thing, and it gives you some very valuable information that can narrow your choices down drastically. Okay, first of all, morphology doesn't change. So once you determine the morphology, that is a concrete detail, a concrete characteristic that you can use to describe this bacteria. Your gram stain result is also concrete, as long as you did the gram stain correctly. Remember, it can be difficult to do a gram stain if you use too much bacteria, if you decolorize wrong, if the bacteria culture is too old, which this one is not. We know this is only 24 hours old. Okay, so your morphology and your gram stain result are concrete characteristics that don't change from a bacteria. Okay, whereas these media, they can be a little iffy. You can have a subspecies of E. coli, for instance, that may ferment lactose where another one doesn't. Okay, so those can be a little more iffy, but morphology and gram stain results are concrete. So if you're ever in kind of a spot where you've got 2 bacteria and you can't determine from all of these results exactly which one it is, go back and look at your morphology and your gram stain result first, because if that agrees with one but not the other, that's probably your correct result, okay? So you would do the gram stain. I'm not going to go over that because you know how to do that already. You would determine the morphology and the gram stain result from that. You can record that in your table. The tables are provided for you. And then you go on to the other types of tests. So the first three, okay, you'll see it's 2, 3, and 4, are media that you are familiar with: an MSA plate, a McConkey plate, and an EMB plate. Okay, you have labeled all these with the type of plate and your unknown and your initials. Remember, we use them lid side down, so we can just pick it up and inoculate it. Okay, so you are going to need your Bunsen burner lit to create an aseptic environment. And then to inoculate these plates, you need a sterile swab. Okay, so remember, these are all selective and differential medias that you have used already. To inoculate, you'll peel your swab, the wrapper down so that you have the handle. Okay, your swab is sterile. Remember, you don't put this in the flame dish just to provide an aseptic environment for this procedure. Pick up your unknown number 1, the tube. Take the lid off, flame the top, go in, moisten your swab, flame the top, put the lid on, and then take that swab and just gently go back and forth across the surface of the outer of the MSA plate. Put that down, so now that's inoculated. And then this can be reused since you are using the same bacteria each time, as long as you didn't contaminate it in some way by putting it down on the table or getting it on your lab coat or something. So you can go ahead and reuse this. If you need a new one, just simply discard it in the proper disposal and then get a new one. But we haven't contaminated this, so we're going to go ahead and keep using it. So you pick up your unknown number 1 again, flame the top, get a fresh sample, in the top, down, pick up your McConkey plate, and just go ahead and gently go across the surface of the agar to inoculate the McConkey plate. Set that aside. Again, you can continue to use this swab. Pick up your unknown, flame the top, get some fresh culture, flame the top, and then inoculate your EMB plate. Same way. Okay, so that is inoculated. So now you're finished with this swab, so go ahead and discard it in the proper disposal bin. Okay, so you have inoculated now your 3 selective and differential medias. Okay, so then we go on to media that we are not as familiar with, or not familiar with yet. So the next one is a starch agar. Okay, and you're looking for whether the bacteria can hydrolyze starch. Okay, so if it can hydrolyze starch, it produces the enzymes amylase and maltase. Okay, if it can produce these 2 enzymes, it can hydrolyze starch, meaning it can break starch down. Okay, so this agar, starch agar, contains starch. And you will be able to tell, which we will go over later, from the results whether the bacteria was able to hydrolyze the starch. Okay, so how do we inoculate this? Well, this requires some heavy growth to see the hydrolysis really clearly. So again, we're going to use a sterile swab. So remove your swab, pick up your unknown. It's unknown number 1, remember we're still doing. Flame the top, moisten your swab, the top. And then for the starch plate, as you'll see in the instructions in your lab handout, you're just simply going to go back and forth on the same line down the middle of the agar several times, so that you're going to have some heavy growth in just the line down the middle of that agar plate. Okay, so go ahead and put that down, and then you can go ahead and dispose of the swab. So now you have inoculated your starch agar plate. Okay, so then, go on to the next page, is gelatin hydrolysis. Okay, so we're using a gelatin deep tube, it's called. So this is a gelatin media. It is the same color as a nutrient broth because it does contain the nutrient broth, and it also contains gelatin, just like you'd see in Jell-O. So it's a semi-solid media, just like Jell-O is kind of semi-solid. Okay, so you're going to make sure you label a gelatin, your unknown number. You have your unknown number 1. For this, you're using inoculating loops, so remember to sterilize your inoculating loop using aseptic technique. Pick up your unknown, flame the top. Remember, this is hot. Cool it on the inside. Get a loop full of bacteria. Top. And then pick up your gelatin tube. Take the lid off, flame the top. Take your loop full of bacteria, and you're going to go right down into the gelatin. Just stab it around in there. It doesn't matter if you mess it up because it's going to become liquid when it goes in the incubator anyway. You just want to get it mixed into the gelatin really well so that it has a good reaction, if it does have a reaction. Okay, and then you flame the top, put the lid on, and you have now inoculated your gelatin tube. And remember to flame your loop. Sterilize it before you put it down. Okay, so you have inoculated your gelatin tube. Okay, that will let you know whether the bacteria can hydrolyze gelatin. If it can hydrolyze gelatin, it can produce the enzyme gelatinase. Okay. So we're going to go on to inoculating the carbohydrates. We are using glucose, mannitol, and lactose. Okay, these are 3 different sugars. Whenever you're describing whether a bacteria can ferment a carbohydrate, you have to specifically say which carbohydrate. Okay, you have to say it can ferment glucose, for instance, or it can ferment mannitol. You don't ever say it can ferment a carbohydrate. That doesn't tell you anything because there's all kinds of carbohydrates. So you have to say specifically which one. Okay, so we are using these 3 for our analysis, glucose, mannitol, and lactose. Okay, they are all a purple broth. This purple broth, it contains the sugar, also contains a pH indicator that will turn yellow if this broth turns acidic. So if the bacteria can ferment the carbohydrate that's in there, for instance, this is a glucose broth, if it can ferment glucose, it will ferment, create an acidic environment, and turn this broth yellow. Okay? So we have a glucose broth, we have a mannitol broth, and we have a lactose broth. So we're going to inoculate each of these simply using our loop and inoculating using the simple technique we used in aseptic technique to inoculate a broth. So sterilize your loop, pick up your unknown, flame the top, remember to cool your loop, get a loop full of bacteria, flame the top, and then pick up your glucose broth, top, take your loop full of bacteria, go in and jiggle it around so you can inoculate that broth, flame the top, put the lid on. You are now finished inoculating that glucose broth. Okay, sterilize your loop, and then go on to your next broth. So you'll pick up your unknown again, flame the top. Remember, this is hot. Cool it. Loop full of bacteria. Flame the top, put it down, pick up your mannitol broth. Take the lid off, flame it. You can take your loop full of bacteria, jiggle it around in there, flame the top, put the lid on. Now you have inoculated your mannitol broth. Go ahead and sterilize your loop. And then go on to the last, the lactose broth. So pick up your unknown again. Top. Remember, this is hot. Cool it. Get another loop full of bacteria. On the top. Down. Then pick up your lactose broth. On the top. Take your loop full of bacteria. Jiggle it around in the broth. On the top. This is your lactose broth that is inoculated now. And then remember to flame this before you set it down, since it has bacteria on it. Okay, so now you have successfully inoculated your 3 different carbohydrates, glucose, mannitol, and lactose. Okay? So then you go on to a hydrogen sulfide test. okay, which uses a SIM media. It's called a SIM media. Okay, this media can be used for other tests. We are only using it for the hydrogen test, okay, hydrogen sulfide test. This is again a semi-solid media, okay, and it's kind of a deep amount of media in the tube so that you can see the reaction clear. All right, so you are going to use a different tool, piece of equipment, okay? It's called an inoculating needle. So this is a lot like your inoculating loop, except it doesn't have a loop on it, okay? It's a needle. You don't ever put a loop on it. When you're provided with a needle, it's supposed to be a needle. Okay, so you want to make sure it's kind of straight as you can get it. Okay, and you're going to be inoculating the SIM media using what's called a stab technique with an inoculating needle. So what you're going to do is, just like a loop, you will sterilize it. Okay, you pick up your unknown sample. Again, cool it like you would a loop. And go in and wiggle it around in there so there's bacteria on the needle. Sterilize the top. Put your unknown down. Then pick up your SIM media. Flame the top. And then what you're going to do is stab this directly down into the media, the SIM media. Okay, what you want to do is get it all the way through the media so that you can get an anaerobic reaction, and you want to get it all the way through the media so that you have a clear positive or negative result. So you're going to stab technique, stab it right down into that media, flame the top, put the lid on, so now your SIM tube is inoculated. Remember, this needle has bacteria on it, and it needs to be sterilized. Okay, so to clarify, SIM media used to determine whether a bacteria can produce hydrogen sulfide or not is inoculated using a stab technique. Okay? So you have your tube. Okay, it's got the SIM media in it. You take your inoculating needle, get some bacteria on it from your unknown culture, and you stab it down into that semi-solid media. Okay, so you're getting it right down into the bottom of the media, all the way up to the top. Okay, so that is a new technique that is used for that type of media. Okay, so then we go on to the urease test. And for the urease test, you're using a urea broth. Okay, it's kind of a little orangey kind of color. So urea broth. If a bacteria can degrade urea, then it produces the enzyme urease. Okay, so the way you'll be able to determine that is to inoculate a urea broth. So again, you go back to your inoculating loop, because you're just doing a simple broth inoculation. Sterilize your loop, pick up your unknown, clean the top, cool your loop, get a loop full of bacteria, clean the top, and then pick up your urea broth, remove the lid, clean the top, go in, inoculate your urea broth, clean the top. You have now inoculated your urea broth. Make sure you sterilize your loop before you put it down. Okay. So again, you're inoculating a urea broth to see if your bacteria can produce the enzyme urease. Okay, so last we have a Simmons citrate agar. Okay, so you have this green colored agar that is in a tube and has been made into a slant by allowing it to solidify as a slant. Okay, so this is a specific type of inoculation too, because this is also something that is an anaerobic reaction. So you want to get it down into the media. Okay, so how are we going to do it? Because this is a solid media. It's not a semi-solid like the SIM or the gelatin. Well, we're going to use a streak and a stab technique. So you're going to need your inoculating loop and your inoculating needle. Okay, so first you're going to go ahead and take your inoculating loop, sterilize it, pick up your unknown, flame the top, cool your loop, get a loop full of bacteria, flame the top, go down, pick up your Simmons citrate outer slant. Okay, remove the top. Remember to keep the outer horizontal, it's easier for you to work with. Flame the top, go in with that loop, and like you learned in aseptic technique, just go along the surface of the outer, just going back and forth and kind of zigzag. Okay, so you have inoculated the surface of the outer now. Flame the top, put the lid on, and then flame your loop. Okay? Then you can set that down. But you're not finished yet. Remember, we're doing a streak and a stab. So now you pick up your needle, your inoculating needle, and you go ahead and you flame that to sterilize it. Okay, pick up your unknown again. Flame the top. Go in, cool your needle, and then jiggle your needle around to get bacteria all over it. Flame the top, put the lid on, get back down. Again, pick up your Simmons citrate agar. Remove the lid, flame the top, and then you're going to stab this right down into that media, all the way to the bottom. Okay, so that you're getting the bacteria way down at the base of that tube. Okay, it's called the butt of the tube. So stab it right down in there, flame the top, put the lid on, and then make sure you sterilize your loop, or your needle, before you set it down. Okay, so in this case, it's a streak and a stab. Okay, so we are doing a Simmons citrate agar. Okay, Simmons citrate agar slant. So the media is at a slant in there, it's an agar. You're doing a streak and a stab. Okay, so streak and stab. Streak and stab. So first you're using your inoculating loop, getting your unknown on it, and going along the surface and inoculating it along the surface. That's the streak part of this technique. And then you go ahead and get your inoculating needle, some bacteria, your unknown on it, and then use that to stab down in. And that is the stab portion of this technique. Streak and stab. Okay, so you have successfully inoculated your Simmons citrate agar. So now we have done all of the media for our first unknown. So you would repeat this whole process along with the gram stain for your unknown number 2 and your unknown number 3. Okay, and then we will wait. We have to incubate these for 24 to 48 hours, and then we will look at how to read the results. Okay? Turn off your Bunsen burner, and we will get these into the incubator, and we will look at